Hello there. Welcome to Fundamentals of Biology, Lesson 7, where we're going to be looking at enzymes. So this is quite a step forward, if you like. So in the previous sessions now, we've looked at the four classes of biological molecules. So carbohydrates, lipids, nucleic acids, and proteins. And what we can now do is start to talk about some of the processes that enable life to exist. And this is something that we're going to explore over the coming sessions, where we take a step up in terms of size in the next session when we're going to look at cells. But today we're going to be talking about enzymes, which are a type of protein that enable a lot of life's processes such as digestion and muscle contraction and nervous systems. All of those things require enzymes to function. We're going to look at today catalysts. So we'll talk about what catalysts are and what they do. And then we're going to talk about enzymes, which are biological catalysts. So they're catalysts that work in life. And then we're going to start looking at how enzymes do what they do. So we'll talk a bit about their structure, and then we'll talk about a couple of modes of action. So a couple of ways in which they work. So we'll look at something called the lock and key hypothesis, and then I'll tell you why it's not accurate. So that's always good. And then I'll tell you about the induced fit model, which is a much more accurate model of how enzymes actually do the things they do. And then we'll go on to talk about how enzymes can be inhibited, so they can actually be switched off. Some of those ways are good and they are a natural part of how life works, but some of them are not so good. So we'll talk about those as well. And as we go through, I'll give you some examples of how enzymes are really, really important to life and how they can actually dictate the geographical distribution of species. And this is particularly important these days as we start to battle the issues surrounding climate change, so warming temperatures around the planet. This is having a big impact on a lot of forms of life, and a lot of that comes down to their enzymes. Let's start by talking about catalysts then. So what are catalysts and what do they do? Well, catalysts are factors that speed up chemical reactions. And this is something that I'd imagine a lot of people know. So if you ask them what a catalyst is, they will know that they speed up chemical reactions. But one thing that is often overlooked is the second point you can see there, is that they exit the chemical reaction unaltered or unchanged. So that's a really important aspect of how catalysts do the things that they do. Because they exit a chemical reaction unaltered, that means they are free to do the same chemical reaction over and over and over again. If a catalyst was altered or affected by the reaction it was speeding up, then it wouldn't be able to do or carry out the same reaction again. So in terms of enzymes, which are a type of catalyst, our bodies would have to just keep producing new and new enzymes over and over again to enable our bodies to function. And as we'll talk about as we go through, this is not really a viable option, because a lot of the chemical reactions that occur in our bodies occur so quickly that the body would soon shut down. So catalysts will speed up chemical reactions, but they exit the reaction unaltered. How do they do that? Well, we can figure this out by looking at this graph. So at first glance, it's not the prettiest thing in the world, but we'll break it down, and hopefully by the end of this slide, you'll have a good understanding of how catalysts work. So catalysts can be inorganic or organic, such as enzymes, but they all work in essentially or in similar ways. For catalysts to actually work then, they do so by reducing something called the free energy of activation. So what that means is, for a chemical reaction to occur, you need a certain amount of energy to actually kickstart the reaction. And that's what's demonstrated in this graph. So let's just have a quick look at it. Let's have a look at our axes first. So along the x-axis, so along the bottom here, we've just got time. So this is the amount of time for a particular reaction to take place. And then along the y-axis up here, we've got energy. So this is the amount of energy that is within a particular system. And then we have two lines here, a blue line, which is an uncatalyzed reaction. So this would be, say, a normal chemical reaction that is at room temperature. And then down here, we have a catalyzed reaction in the red line. First off, let's forget about the red line. So forget about that one for now. Let's just talk about this blue line here and have a look at what it tells us. So the first thing we can see is that we start here on the left, and let's actually use an example here because it might help. So if you have a gas stove at home, a gas cooker, or say we're in the lab and we turn on a gas tap, well, out of that gas tap comes methane. So if you remember methane from our very first session, methane has the molecular formula CH4. It's a carbon atom surrounded by 4 hydrogens. Now, that is natural gas, or it's a main component of natural gas. And we can make that react. But if you were to turn your oven on, or you turn the gas tap on in the lab, we are about here. What does that methane do? It doesn't do anything. You just turn that gas tap on and the methane will just leak out into the room and smell pretty bad. However, if you want to create a chemical reaction, what do? You have to add energy to the system. So we do that by adding a flame or adding a spark. And what that flame will do is it adds energy. So we can see energy along the y-axis here. And what that does is it adds energy, which is represented by this upward line here of the graph, until we get to the top up here. And then we start curving back down again. The top of this graph here, the top of this line, is where the chemical reactions start to occur. And this is what we call the transition state. So you can see it down here, the transition state. That is where bonds are broken and new bonds are formed. So in the case of our methane, so we think about it, it's CH4, so carbon surrounded by 4 hydrogens. What will happen if there's not enough energy, so we're just at room temperature and we turn our gas tap on, the methane will just leak out into the room. We add a flame to it, that adds energy to enable the methane to get up to the top of the graph here, so for it to reach the transition state. And what will happen then is those hydrogens and carbons will start to break apart, and then the oxygen that is in the air around us will start forming new bonds with the carbon and the hydrogen. So oxygen will combine with hydrogens to make water, and oxygen will form with or bind with the carbons to make carbon dioxide. And this is essentially combustion. This is where a lot of the carbon dioxide in the atmosphere comes from. It comes from burning fossil fuels, which are largely carbon and hydrogen. And when you break those hydrogens off of the carbons, and oxygen will combine with the hydrogens to make water, and it will combine with the carbons to make carbon dioxide. And as those new bonds are formed, so as the CO2 and water are produced, we can see this downward slope on the graph, and this represents the release of energy. So in the case of our methane, that energy is being released as heat and as light, and also as some as sound. We add a flame to our methane, that once there's enough energy, we reach the transition state where the bonds between the methane start to break apart, and then the new bonds to make carbon dioxide and water start to form, and that releases that energy in the form of heat, light, and sound. So that's basically time frame, if you like, or of a typical chemical reaction. Now, one thing that we need to talk about is this upward part of the graph. Now, because this requires, for this chemical reaction to occur, it requires that certain amount of energy for the reactants. So in this case, we could say that it's methane and oxygen down here. For the reactants to start reacting to reach the transition state, they have to have this much energy. So this requirement of energy here acts as a barrier. Without that energy, the reaction does not take place. So we actually call this the activation energy barrier. So the amount of energy that is required to reach the transition state. If you think about it, that's actually quite a good thing that this barrier exists, because otherwise all chemical reactions that could occur would occur instantaneously, and we would all we'd all go up in a bunch of flames and smoke and sound and heat, which wouldn't be very good for life to continue existing. So fortunately, we have this barrier, this activation energy barrier, that reactants have to overcome to reach the transition state. Now let's take another example now. Say you had a cup of coffee and you put a spoonful of sugar in your coffee. Well, the sugar in that coffee, so if you remember from our previous sessions, the sugar that we would put in coffee is sucrose, so it's a disaccharide. Well, that disaccharide needs to be broken down into glucose and fructose so we can use it. So typically, if I took a beaker of room temperature water and I was to stir a spoonful of sugar into it, that sucrose and water would be down here. And it would sit there for years and years and years, and nothing would happen to it. For that sugar to break apart, to hydrolyze into glucose and fructose, it needs to reach the transition state for the chemical reactions to occur, for it to break apart. Clearly, we're not going to be able to do what we did with our methane, which is to heat it up. Every time you eat something or you consume a cup of tea or something, you're not going to lie over a Bunsen burner and heat your stomach contents up so the contents can reach the transition state. That would be highly inconvenient and would result in a mild case of death. So not particularly useful. So instead, what we need is to reduce the amount of energy that is required for those reactants to reach the transition state, so the chemical reactions can occur with a lower amount of energy. So basically, we're lowering or reducing this activation energy barrier, and that is where catalysts come into it. Catalysts. So we can start looking at this red line now, this second line, showing a catalyzed reaction. What we can see if we look at this is that the transition state is much, much lower. And what this means is, particularly in terms of enzymes and chemical reactions that occur in our bodies, it means that chemical reactions can now occur at our normal body temperature. So for humans, that's typically between about 36 and 38 degrees Celsius. So a catalyst will lower or reduce the activation energy barrier, reducing the amount of energy that is required for the reactants to reach the transition state. So it's an important thing to remember or to realize that enzymes do not add energy to a chemical reaction. They reduce the amount of energy that is required for that reaction to take place. So I know that's quite a bit to go through, so you might need to go over that a couple of times and to listen to this part of the video again. That is essentially how catalysts work. They lower the activation energy barrier, therefore reducing the amount of energy required for reactants to reach the transition state. How they do that is something that we'll look at in a bit. So that's what we've just talked about there is true of all catalysts, whether you have an inorganic catalyst such as manganese dioxide, which can hydrolyze hydrogen peroxide to water and oxygen, or whether you're looking at a biological catalyst such as an enzyme like catalase, which will catalyze or hydrolyze the same molecule. So catalase will break down hydrogen peroxide in the body down into water and oxygen. And we'll talk about that one again in a bit. Enzymes then are biological catalysts. They speed up biological reactions. Those are chemical reactions that occur within living organisms. And again, it doesn't matter whether we're talking about an individual bacterium or whether we're talking about a human or whether we're talking about a mushroom or a oak tree. So all forms of life are completely dependent on enzymes to speed up the chemical reactions, because otherwise they just would not happen quickly enough because of that activation energy barrier we just talked about. So here's the example that I gave you on the previous slide then. So as an example, sucrose could break down to fructose and glucose, but at room temperature or normal room temperature, it would take years and years, maybe decades to do so. However, we can add the enzyme sucrase, so we can see there that most enzymes end in ase, but not all. So just be aware of that. There are a lot out there like pepsin that don't end in ase. But a lot of the ones that we have talked about in previous sessions, so sucrase, maltase, lactase, amylase, these are all examples of enzymes that do end in ase. So if we add a bit of sucrase to our beaker of sugar water, what we will see is that the sucrose will be hydrolyzed to glucose and fructose almost instantly. It happens very, very quickly. And we can actually see that chemical reaction in the diagram below. So here we can see our sucrose, so glucose and fructose. There's our glycosidic link that was formed by through dehydration synthesis. We're going to do hydrolysis, so we need to add a molecule of water. And then here's our enzyme. Here's sucrase that's actually going to facilitate or catalyze this reaction. And then here we can see the products, as we call them. So when we talk about an enzymatic reaction, what goes into the enzyme, so in this case sucrose, is called the substrate. And I've got this on a slide in a sec, but the substrate goes into the enzyme and then the products leave. So in this particular reaction, we've got the enzyme sucrase. Sucrose is going to be the substrate. The chemical reaction will occur, in this case hydrolysis, and then our products, which are glucose and fructose, will leave. And that will happen almost instantly once we add some sucrase. Our only other option really to start hydrolyzing our sucrose to glucose and fructose would be to really heat up that sugar water, to get it boiling, to add energy. And maybe we could speed it up by adding some acid, some hydrochloric acid, to help to chemically hydrolyze that glycosidic link as well. Alternatively, we can just add some sucrase, and that will do it very, very quickly for us. Okay, so let's start having a little look at the enzymes then, and we're going to go in and talk a bit about their structure and how they do the things that they do. So one really important thing to know about enzymes is that they are very, very specific. So if you remember from the previous session where we looked at proteins, we talked about the fact that proteins have a very specific structure, and that structure equals function. So their shape and size will determine the job that they can do. And enzymes are proteins. The vast majority of enzymes are proteins. There are a few made of RNA, but the vast majority of them are proteins. And just like other proteins, they are made of a long chain of amino acids that is highly coiled and folded to give them a very specific structure. So what this means is that enzymes are specific. They will typically only work on one specific reaction. So the example that we talked about just now, so sucrase has one job in life, and that is to break apart or to hydrolyze sucrose into glucose and fructose. If you wanted to build sucrose from glucose and fructose, you would need the enzyme sucrose synthase. Now, we don't actually produce that enzyme because we're not plants. Plants will actually produce that enzyme because they need to take the glucose and the fructose, join them together through dehydration synthesis, so they can then transport those sugars around their bodies. So plants will produce the enzyme sucrose synthase to do that. But when we consume that sugar, that sucrose, we need the enzyme sucrase. So we need specific enzymes to carry out specific reactions. And this supports that. So they will often have a preference of one direction of chemical bond making or breaking. So again, in the case of sucrose, We have sucrose synthase that will actually build it, so dehydration synthesis. And then we have sucrase that will hydrolyze it, so it will break it into glucose and fructose. So enzymes are very specific in what they do. And again, that comes down to their structure. And they have a range of sizes, so they are just the right shape and size to do the job they need to do. So one of the smallest that we know of is only 62 amino acids long. So it'll have 62 amino acids in its primary structure. And that one is called 4-oxaloocrotonate tautomerase. And then one of the largest that we know is about 2,500 amino acids long. And that one's called fatty acid synthase. So we have many, many enzymes that carry out a whole range of different functions within our bodies. And different species will often have different enzymes to enable them to do what they do. They will be perfectly suited to their job. What we also find, and we're going to talk about this more in a sec, but they will have very, very specific temperature and pH ranges. So hopefully you can remember why that will be based on what we discussed in the last session on proteins. So keep remembering that enzymes are proteins. And we talked in the last session about what happens to proteins when they are exposed, particularly to high temperatures, but also when they're exposed to the incorrect pH. So what will happen to them? You're absolutely right. They will denature. They will unravel. The bonds that hold together the secondary, tertiary, and quaternary levels of structure will unravel, and that will basically mean that the enzymes will no longer be able to do their jobs. And this is one of the reasons why life tends to try really hard, life does try really hard to stay within optimal conditions in terms of temperature and pH. So the last thing to know about enzymes then is that they react very, very quickly. We're talking something called femtoseconds. So that to me just means mind-bendingly quickly. To give you an example of this, got an enzyme that I've mentioned previously called catalase. Now, catalase is an important enzyme in our cells, and we'll talk about particularly where they're found in a later lesson. But catalase is an important enzyme in our cells because it breaks down a really toxic compound that is a waste product of a cellular process, and that waste product is called hydrogen peroxide. You may be familiar with that. People have used it for years to dye their hair, to bleach their hair. It used to be used extensively as a disinfectant. It's a really nasty substance, and it will sort of tear apart our cells if it's left to its own devices. So we have to have an enzyme in our cells that will break that hydrogen peroxide down very, very quickly. And it will do so at an incredible rate of about 10 million reactions every single second. So catalase will be able to break down hydrogen peroxide into water and oxygen at a rate of about 10 million reactions every second. So mind-bogglingly quick. Okay, so those are enzymes, very, very specific. Let's talk a little bit more about the optimal temperature range then, because it has such an important fact, and it's such an important impact on life. And what we can see here in this diagram, so let's start by looking at the axes. So along the x-axis here, we have temperature in degrees Celsius. And then along the y-axis, we have the rate of reaction. So this refers to how quickly the enzyme is carrying out its chemical reactions, how quickly it is turning or converting substrate into product. And what we can see here is we've got a black line and a red line. The black line is the reaction profile for a typical human enzyme, and the red line is a temperature profile for an enzyme from a heat-loving bacterium. So that's called a thermophilic bacteria, so a heat-loving. Forget about that one for a moment. Let's focus on the black line, so the human enzymes. And what we can see here is that as temperature increases, the reaction rate also increases. So the enzymes start to work quicker and quicker. And this is because as the temperature increases, molecules start to move faster. So that is essentially what heat is. Heat is a measure of kinetic energy, how quickly things are moving. So as things start to move up in temperature or increase in temperature, they start moving much more quickly. And what that means is we start to get more and more collisions between the enzyme and the substrate. So we start to get an increase in reaction rate. And then we get to the top of this curve. And so we would refer to this as the optimal temperature for our human enzymes. This is when the enzymes are working at their fastest. So the optimal temperature gives us the fastest reaction rate. And as you can imagine, that optimal temperature is the temperature that we find in humans. So typically, as I mentioned earlier, that's about 36 to 38 degrees C. So if we follow this top of this curve down, we can see that we're at about 36 to 38 degrees Celsius. Notice what happens if we keep going above that temperature though. We get a very drastic drop off in reaction rate. Why do you think that is? We mentioned it just now. You're absolutely right. It's because the enzyme is denatured. Once you go above the optimal temperature range, so the temperature range that we find within humans, the enzymes will start to denature, they start to unravel, so they are no longer able to do their job, and you'll get a very, very quick drop off in reaction rate, ultimately leading to death of the organism, in this case, humans. And we can see this with something that we experience called fever. So a fever is something called a systemic immune response. This is a very serious body-wide immune response that the body will trigger when it is having a particularly hard job getting rid of a pathogen. So a pathogen could be a virus or a bacterium. Essentially, if the main immune response, so using things like white blood cells, if that doesn't work, what will happen is the body will increase its thermostat. So the thermostat for us is located in a part of our brain called the hypothalamus. And the hypothalamus is responsible for keeping us at a nice 36 to 38 degrees C. But what it will do to trigger a fever is it will increase that. So now it might increase it from 37 to 39 degrees C, or maybe 38 to 40 degrees C. But what you can see is as we start to increase in temperature, the reaction rate starts to drop off. So you might think, why would the body do that? Well, it does it in an attempt. It's essentially a kill or cure approach. So what the body is trying to do by triggering a fever is it's trying to denature the proteins and denature the enzymes within the pathogen that's infecting us. So it's basically trying to kill off the bacterium or the virus that is infecting us. The downside to that is at the same time, it's actually killing us. So that's why I say it's a kill or cure. The hope is that the raised temperature will kill off the pathogen, and then the temperature can return back to normal and you can recover from that fever. But it's one of the reasons why a fever is such a dangerous time, is because the temperature is raised, the reaction rate of the enzyme starts to drop off quite quickly. And that's why people that are suffering from fever, their body is essentially starting to break down. You start to see things like hallucinations and uncontrollable shivering, and the muscles and the nervous system just aren't functioning properly because the enzymes are starting to denature. Let's move over to the red line then. What we can see here is the temperature reaction profile for a thermophilic or heat-loving bacterium. So these belong to a group of organisms that we would call extremophiles. These are organisms that live in extreme environments. So you could expect to find this kind of bacteria living in around a hydrothermal vent at the bottom of the ocean, or in a caldera of a volcano, or in a hot spring in somewhere like Yellowstone National Park. And what we can see is that the shape of the profile is the same as for the human enzymes. It's just set at a much warmer temperature. So for these guys, their optimal temperature, so where their reaction rate is the highest, is at about 78 degrees or 76 to 78 degrees C. Well, we would have died a long time ago at that temperature. So yeah, we're probably dead if we spend any amount of time at, above 40 degrees C. Then we really don't last very long at all. We're sort of going into the minutes realm. Whereas these guys are nice and toasty and operating at their highest level at about 76 to 78 degrees C. But the same relationship with temperature exists. If they drop in temperature, so the temperature gets lower, the reaction rate will get lower as well. Now, this isn't because the enzyme is denaturing at lower temperatures. It just means that with the lower temperature, you get a lower amount of kinetic energy. So the substrate in the enzymes are moving that much more slowly that you're getting a reduction in collisions between the substrate and the enzyme. So therefore, the reaction rate will drop. But if they go above their optimal temperature, you get the same rapid drop-off in reaction rate, and that is because of denaturation. The enzymes will denature. That's essentially what we can see across all forms of life. They will have a particular suite of enzymes within their bodies that have an optimal temperature for them to function. And those organisms will need to be within that optimal temperature. So for cold-blooded organisms, so whether we're talking about something like a lizard or if we're talking about something like a fungus, because their internal body temperature is determined by the external temperature or the environmental temperature, they need to be within a particular habitat or a particular environment where they can stay within their optimal temperature range. And there's some interesting ways that organisms deal with this. So one of my favorite examples for this is the common carp. So we get common carp in this country in lakes and ponds, and you might keep carp if you've got a fish pond at home. You might have some relatives of the carp, so goldfish or koi carp, in your pond at home. And what you can notice is that those carp will be perfectly at home living in that pond in the middle of winter when the water temperature may be close to zero. It might be down to 3 or 4 degrees Celsius. And the carp will survive quite happily in there. Their metabolism is very low and they don't eat at that temperature, but they will survive. And then they will also be particularly happy or very happy in the middle of summer when the water temperature is 28 to 30 degrees Celsius. So how is that possible? If we look at the graph here, we're looking at a really wide range of temperatures. Well, they will actually do that. They survive these temperature changes through something called acclimatization. And essentially what they will do is they will produce new enzymes that operate at the temperatures of the coming season. So a carp in a pool in the middle of winter, so where it's 4 or 5 degrees Celsius, they have a suite of enzymes that are suitable for working at those cool temperatures. But as they start to move out of winter and into spring, certain genes will switch off and other genes will switch on. And those genes that are switched on will start to code for new enzymes that operate at warmer temperatures. And this process carries on slowly through spring, and by the time you get into summer, they will have a whole new suite of enzymes that work at the new warmer temperatures. And then as they move from summer into autumn and into winter, the same process happens in reverse. Those summer genes will switch off, the winter genes will switch on, and then by the time the cold water comes in, they will have the new suite of enzymes that operate at those colder temperatures. But it's important to realize that they have to go through this process of acclimatization. If you were to take a carp from 5 degrees Celsius water and just stick it into a fish tank with 30 degrees Celsius water, they would die very, very quickly. They need a period of weeks and sometimes months to swap over and to change their suite of genes to their suite of enzymes that operate at the new temperatures. So life is very adaptable, and evolution has driven this ability to function. But it's important to realize that not all animals have this ability, or not all life forms have this ability. So if you take something like a clownfish, for instance, so you'll all be familiar with Nemo, common clownfish, because they live in tropical areas, so they live in around tropical coral reefs, where the temperature is very, very stable, so typically around 25 to 26 degrees Celsius, they don't experience the same seasonal changes or fluctuations that something like the carp does. So they never evolved the ability to produce these different enzymes that operate at different temperatures, because they never had to. They've always experienced the same temperatures. And this is part of the reason why we're starting to see certain issues around the globe. So if we look at something like coral bleaching, well, corals, like clownfish, have evolved to live at these in these tropical oceans where the temperature has remained fairly stable for thousands of years. So they've never had to evolve the ability to deal with these fluctuating temperatures. And now that we're starting to see rising temperatures in the ocean, then a lot of them are unable to cope, so they will actually bleach. So temperature is really important to enzymes, and organisms will do an awful lot to maintain their optimal temperatures, or to stay within an environment that has their optimal temperatures. As mammals, so we're mammals, we're warm-blooded animals, and our bodies work very, very hard to maintain our temperature within the 36 to 38 degrees Celsius range. And we'll talk about how we do that in a later lesson. I've kind of just described this a little bit using the clownfish and the coral and the carp, but here we've got another example, and it's using a very exciting type of animal, the barnacle. I think, if I remember rightly, Charles Darwin spent about 7 years studying barnacles, so they must be quite special. But what we can see here then are two species of acorn barnacle from the UK, so native to our shores, and it gives us a nice indication of how temperature will affect the geographical distribution of species, but particularly cold-blooded species, because remember, they are completely dependent on their environmental temperature to control their internal temperature. So if we look at the left-hand part of this diagram, then we can see this species, and we can see that their distribution is limited to the west and southwest coasts of the UK and Ireland. But they will go all the way down to the far southwest of Ireland, and also down to the Isles of Scilly. But we don't find them on the east coast of the UK. And a lot of that, or it all comes down to temperature. So the west coast of the UK and Ireland is basically at a warmer temperature because of the Gulf Stream. So if you've not heard of the Gulf Stream before, it is a system of ocean currents that bring warm water up from South America, and they sort of go past our west coast, bringing the warmer water with them, which also warms the air above the water, which is part of the reason why we have such a balmy climate in the UK, on the west coast of the UK, particularly down here in the southwest. So this species requires or prefers these warmer waters that are produced by the Gulf Stream. But over here off the east coast of the UK, we get much cooler waters, so this species doesn't exist here. If we look at this species, well, their optimal temperature is just a slight bit lower than the other species. So it enables them to move into the cooler parts of the UK, so they can survive on the east coast. They can also survive around the west coast, but you don't find them as prevalent on the far southwest of Ireland, and also the far southwest of Cornwall down here, and you also don't find them around the Isles of Scilly, which are just down here. And that's because they're unable to tolerate those slightly warmer temperatures that we find there. So here we can see how temperature affects the geographical distribution of these two species. And the reason that this is becoming such a big issue for us regarding global warming or climate change is because you can look at these diagrams and you can start to imagine what could happen in the future if our oceans continue to rise in temperature. So let's take a look at this species again on the left here, Well, let's say we increase our water temperature, our average water temperature around the UK by 2 degrees C. Well, this species is now going to be able to move its distribution, extend its distribution around to the east coast. It may get to a point where it's too warm on the southwest, so here its habitat will start to shrink. So it's gaining habitat here, but it might be losing habitat down here. This is now going to bring it into competition with this species. So up until now, this species doesn't have to share its habitat on the east coast. It's got that to itself. But as the temperatures rise, it's now going to be facing competition on this side. But also, not only is it facing competition along the east coast, as the temperature rises, it already cannot stand the warmer temperatures on the far southwest. So as the temperatures start to rise, its habitat is going to start to shrink. Its distribution will start to move away from the western coast of the UK and Ireland. It might be, will get to a point where it doesn't exist at all on the west coast, just like this one at the moment doesn't exist on the east coast. So now this species has lost a large part of its habitat, and it's also in competition in parts where it wasn't before. So what would this species do? Well, we're starting to see this all across the planet now, with loads of species, not just animals, but with plants and bacteria and fungi. They are starting to migrate. They're starting to move away from the equator and they're starting to move north and south towards the poles into cooler temperatures where they will find the temperatures needed for their enzymes and proteins to function properly. So it might be that this species starts moving further north up towards Scandinavia or somewhere like that. So we are starting to see this gradual march, this gradual migration of species in the face of climate change. Those species that cannot migrate, that cannot move, well, they are relying on the movement of their young. So barnacles will produce a larvae that can actually move around in the currents. But if you've got species that don't have that, that don't move, well, they've got two choices. They can adapt, so they can start producing enzymes that can operate at the new temperatures, or they die, they become extinct. So this is one of the reasons why climate change is such a big issue. And using two examples of barnacles. On this slide, we can see pH and the role that pH has on reaction rates. Very similar graph to the previous one that we looked at with temperature. But instead of temperature, we have the pH scale along the bottom. So hopefully you know, but the pH scale measures how acidic or basic a solution is. So pH of 7 that we can see over here is neutral. So that is the sort of equal amount of hydrogen ions and hydroxide ions, as we refer to it. If you drop below pH 7, you become acidic. And as you go above 7 up to 14, that is basic. And typically, life will exist ideally in a pH of about 6 to 8. But there are examples where we see extremes. So we can look at a couple here. And again, up our y-axis here, we've got the rate of reaction. So this is how quickly the enzyme is carrying out the chemical reaction. And what we're looking at here are actually two human enzymes. So these are enzymes that are in us working away right now. And these are two enzymes from our digestive system. On the left-hand side here, with the black line, we have the reaction rate for an enzyme called pepsin. And pepsin is an enzyme that it lives in our stomachs, so it exists in our stomachs, and it starts the breakdown of proteins that when we eat our food. And as you can imagine, because it exists in our stomachs, where we produce a lot of hydrochloric acid, it needs to be able to survive in very low pHs. And that's exactly what we find. We see that its optimal pH, the pH at which its reaction rate is the highest, is about the pH of 2. And that's the pH that we typically find in our stomachs. But notice that this line is much more symmetrical than what we saw with the temperature graph. So remember, with temperature, as you drop below the optimal temperature, the reaction rate just slows down because of a reduction in collisions between substrate and enzyme. As you go above, you get a rapid drop off because of denaturation. With pH, the reason that we've got a symmetrical shape here to this graph is that if you drop below or above the optimal pH, the enzyme will denature. So it will denature at above or below the optimal pH. So it's very important that the enzyme remains within the correct pH. Now, if we start moving or carry on the journey of food from our stomach, it goes into the small intestine. And what we can see here is that we have another type of enzyme called trypsin. Now, trypsin is an enzyme that works in the intestines. And what happens to the stomach contents, it produces something called chyme. And chyme is very, very acidic. It moves from our stomach into our intestines, where the acid gets neutralized by things like bile salts, so produced by the gallbladder. And what that will do, it will raise the pH of the intestines to about 8. The enzymes that we find, so trypsin, which lives in our intestines, operates most effectively at a pH of about 8, which we can see here. That's where its highest reaction rate is. But the same rules apply. It has the same pattern. You go below a pH of 8 or above a pH of 8, the reaction rate drops off because of denaturation. Hopefully this illustrates why enzymes have such a specific temperature and pH that they need to exist in to operate properly, and why certain things like climate change are affecting it. pH we're also starting to see some issues with in the natural world, particularly again relating to coral reefs. You might be familiar with something called ocean acidification, and this is where, as CO2 levels continue to increase in the atmosphere, though that CO2 is starting to dissolve into our oceans. And as CO2 dissolves in water, part of that CO2 turns into something called carbonic acid. And as it's an acid, it starts to lower the pH of the ocean, and that is also attributing or contributing to the bleaching of corals. And it also has an effect on the fact that corals, and they struggle to build their skeletons, and not only corals but other marine invertebrates. They're unable to build strong skeletons in a lower pH, and shells, I should say. So temperature and pH have a really big impact on enzymes. So how do enzymes do what they do then? How do they actually speed up these chemical reactions that we've been talking about? In an enzymatic reaction, I've mentioned that the substrate is what goes into the enzyme, and the reaction occurs and then the products leave. So in an enzymatic reaction, the substrate binds to something called the active site of the enzyme. And we can see that down here on this diagram. So the grey part here is the enzyme. The part at the top here with the shape to it, that is called the active site. And the active site is the part of the enzyme where the chemical reaction will actually occur. So the reactant that the enzyme acts on is called the substrate. So we can see here that the substrate is this green shape here. The substrate will come in and it will attach to the enzyme at the active site. Once they are bound together, we have something called the enzyme substrate complex. So that's what we can see here. So that's when you now have the enzyme with the substrate inside the active site. Once that is together, the chemical reaction will occur, and then that produces something called the enzyme products complex. So you can see here that they've done a change in color, but we can think of it as the sucrose hydrolyzing into glucose and fructose, and then the products will leave. So once the reaction is completed, the products leave, and then that frees up the enzyme to start the reaction all over again by binding to the next substrate molecule. So the business end of the enzyme is called the active site that you can see here. Substrate comes in, the reaction occurs, and then the products leave. How does that work then? For a very long time, when people talked about enzymes, they talked about it in relation to this lock and key hypothesis. You can see here that it was came upon by Fischer in 1894, so a long time ago. What they discovered back then was that the substrate appears to fit into the enzyme like a key into a lock. So that's to say that the enzyme active site is just the right shape and size for the substrate to fit into. And that was great. And it kind of still does hold up a little bit. You still hear that you'll still hear people refer to the lock and key hypothesis or the lock and key model in terms of how enzymes work, because it's kind of true. The substrate and the enzyme do match up. The substrate fits nicely into the active site. But it's not accurate in the sense that it still doesn't tell us how the enzyme does what it does. How does an enzyme lower the activation energy barrier to enable the reaction to occur? All it tells us is that the substrate fits into the active site. We needed a better model, a more detailed model. And that came about in 1958 by Koshland, and it's called the induced fit model of enzyme action. And what we can see here, we look at this diagram. So the blue part is the enzyme. We can see the active site up here. And then this little red thing up here, this is our substrate molecule. So what will happen is the substrate will come along and it moves into the active site. And then something happens. And that is the enzyme will change shape around the substrate to make the enzyme substrate complex. The enzyme it's changing its shape to wrap around the substrate. And what we're going to do next is look at why that's important, because what this actually does is it gives us some ideas, it gives us some clues as to how enzymes actually carry out or speed up these chemical reactions. So induced fit allows the enzyme to act as a catalyst. And we know that it does this by lowering the activation energy barrier. And it does it through 4 different modes of action. So the first thing that induced fit does is it provides a template for the substrate. And what I mean by this is that the active site is just the right shape and size to only allow the substrate in a very, very specific order or in a very specific direction. So if we go back to, let's go back a second to all the way back. Where's my sucrose? Okay, so let's take glucose and fructose again over here that we've talked about before. We know that dehydration synthesis is going to occur between these 2 hydroxyl groups. So we're going to remove that water molecule to make a glycosidic link between that oxygen there and this carbon. We need, when these 2 go into the enzyme, these hydroxyl groups, we need them to be facing each other. We can speed up the breaking of these bonds and the forming of that new glycosidic bond. If they came into the active site in a completely different direction, so if this hydroxyl group was pointing over here and this one was pointing up here, well, that's not going to be conducive to that chemical reaction occurring. So when I say that induced fit provides a template for the substrate, what I mean is that they will only allow the substrate in a very specific order that facilitates the making and breaking of specific bonds. So in this case, it would be glucose and fructose would be allowed into the active site with these 2 hydroxyl groups facing each other. The second one is that as induced fit occurs, as the protein, so as the enzyme actually changes shape to wrap around this substrate, it will distort it, it will twist it and put pressure on it. And what that will do is it will speed up the breaking of bonds. And then in turn, it will help the making of new bonds. So by changing shape, it's physically distorting and twisting the substrate. The third one is that once induced fit has occurred, if we look at this diagram, we can see now that inside the active site is basically like a little cocoon. You've got an area in here that is separated from the area out here. And what that means is you've got a separate little environment in here. And depending on what amino acids you have making up the active site, so remember, an enzyme is just a protein. A protein is made up of lots of those different 20 amino acids. And those different amino acids have different properties based on their R group. So some will be acidic, some will be basic, some will be hydrophobic, some will be hydrophilic. So depending on what amino acids make up the active site, this can now be, say it's an acidic environment or it's a hydrophobic environment, whatever environment you need to speed up that particular chemical reaction. So by induced fit, we get to produce a microenvironment. The fourth one is that we can get some temporary interactions between the enzyme and the substrate. During the making and breaking of bonds, parts of the chemical reaction can occur using the active site. So we can see here actively interacting in the reaction in a reversible manner. So why is that important? This bit, it has to be in a reversible manner. So why does any interaction between the substrate and the enzyme have to be reversible? Again, I think I can hear you say it. It's because of the second rule of Enzyme Club. If you haven't seen Fight Club, that will make no sense to you. But if you have, we know that the first rule is that catalysts are factors that speed up chemical reactions. But the second rule is that they exit the reaction unaltered or unchanged. Okay, so we've got two rules of catalysts and therefore enzymes. They speed up chemical reactions and they exit that reaction unchanged or unaltered. Any reaction that we get between the substrate and the enzyme has to be reversible, so the enzyme exits the reaction unaltered. So those are the four different modes of action by which enzymes are able to lower that activation energy barrier to actually act as catalysts to speed up the chemical reactions. So that's quite a lot to take in. If this is the first time you've ever talked about enzymes, you could just sort of make a note of those things and go back over them after this session. And by the time you've been through them a few times, hopefully it will start to sink in. So we've seen how important enzymes are, and we've talked about some examples of as we've gone through. But we have thousands of enzymes within our bodies carrying out various chemical reactions. And it's important that they aren't all going on all of the time, sort of left to their own devices. There needs to be an element of control of enzymatic activity. And therefore, we have this control of enzymes, and we can do this in a few different ways. The first one is compartmentalization. And this is it just makes efficient sense, because you don't necessarily need all enzymes in all parts of every cell throughout the body. You need specific enzymes that do specific jobs in the parts of the cell where they're needed. Some parts of a reaction may take place in different parts of the cell. You can see a generic animal cell. If you've never seen one of these, don't worry. We're going to start looking at these in the next session. So we'll start looking at cells. But what we know is inside the cell, there are lots of different organelles or lots of different machines inside the cell. So if you are enzymes that take place in something called respiration, well, then you'll be down here in these little things called the mitochondria. If you are involved in something like fatty acid synthesis, well, you'll be over here in the smooth endoplasmic reticulum. So different parts of a reaction that require different enzymes will have those enzymes, but we aren't just a random soup of all enzymes everywhere. So compartmentalizing them is important. The next thing we can do is we can actually regulate them. We can switch enzymes on and off using things called inhibitors and activators. So an enzyme typically exists as you can think of it as a vibrating lump, if you like. And if you think about the active site, when it's the right shape to accept the substrate, well, that is in its on position. But if you change the shape of that active site, well, then it's in its off position because the substrate cannot get in. And the enzyme is constantly vibrating between that on and off position. And we can stabilize it using inhibitors and activators. So if you have an activator, which is a molecule that will bind to the enzyme, that will stabilize it in its on position so the substrate can get in. If you join an inhibitor to the enzyme, Well, an inhibitor will change the shape of the active site and stabilize it in its off position, so the substrate can no longer get in. And this is a process that we call allosteric control, the switching on and off of our enzymes. And what we often find is that the products of an enzymatic reaction will act as the inhibitors or the activators. It will act as the allosteric controllers, if you like. So if we think of an example that we've already talked about today, so say you have a particular sweet tooth. So you have a cup of coffee, and instead of putting a sugar in your coffee, you put 4 spoonfuls of sugar in your coffee. Well, suddenly you get a huge influx of sucrose into your body. Now that sucrose needs to get broken down into glucose and fructose, but it also needs to be absorbed by the intestines into the blood, and it needs to get dealt with. So what happens is your enzyme sucrase will set to work breaking sucrose down into glucose and fructose. But it's quite possible that you can get to a point where you've got a backlog of sucrose and glucose and fructose that the body needs to deal with before you carry on the reaction. So the products of the reaction, in this case, will be glucose and fructose. Well, they can actually attach to the enzyme sucrase and stabilize it in its off position. So they switch it off temporarily whilst the body deals with the build-up of the glucose and the fructose. Once the body has dealt with them and they've used them up and they've been absorbed into the bloodstream, the inhibitors, in this case the products, will leave the enzyme and switching it back into its activated form, into its on the stage, and then the whole process can carry on again. So this is the process that we call allosteric control, where the products will actually regulate the enzymatic rate by inhibiting the enzyme, turning it off for a spell, and waiting for the body to actually deal with the buildup of the products. Enzymes can be controlled through compartmentalization, but also through allosteric control, the switching on and off of the enzyme. We can also find other types of things inhibiting enzymes, so switching them off. So we can see, we just mentioned that some of this is a natural part of life, so natural part of what happens in bodies and in cells. So enzymes can be switched off when they need to be. But we also find that drugs and toxins will also inhibit our enzymes. And they will inhibit our enzymes through two different modes. One is called competitive inhibition, and the other is called non-competitive inhibition. And we just mentioned this, that this can also be a part of allosteric control. But we're going to go through and focus a bit more now on the drugs and toxins. And you can see down here, this diagram shows a normal, healthy functioning enzyme. So here's the enzyme with its active site. Here's the red substrate. So the substrate will come in and it will bind to the active site where the reaction will occur. What we can see over here on this part of the diagram is something called competitive inhibition. So this little yellow thing here is a competitive inhibitor. And if we move on to the next slide, we can see that competitive inhibition is where a similarly structured compound or molecule competes for the active site of the enzyme. We can see that in this case, the inhibitor, it's just a molecule or a compound that can actually get into the active site and bind to it. And when it's in there, it prevents the proper substrate from getting in there and the reaction taking place. So competitive inhibition it can be a real problem because it will essentially drastically slow down the reaction rate of an enzyme. So I put a few examples here. So one of the most important discoveries in human history, arguably, is antibiotics. And antibiotics basically kill off bacteria, and which is particularly useful for fighting bacterial infections. And the first antibiotic that was discovered was penicillin. And penicillin works as a competitive inhibitor by inhibiting enzymes in certain bacteria And those enzymes are responsible for producing the cell walls of those bacteria. So you inhibit those enzymes with your penicillin. The bacteria are no longer able to produce their cell walls, so they're no longer able to grow properly and then reproduce by dividing. So instead, they kind of just grow and then they'll just die off. They'll split open. So penicillin works as an antibiotic by competitively inhibiting the enzymes. And this is how a lot of our drugs, medical drugs that we use, actually work. They work by targeting specific enzymes. So I'll give you a good and a bad example here. So the good example is Viagra. I hope you're not sniggering. But Viagra is an important one to discuss. It works by competitively inhibiting enzymes that break down a neurotransmitter called nitrous oxide. And nitrous oxide will bind to receptors in smooth muscle tissue, and it basically helps to dilate blood vessels. But then what this enzyme does is it will break down the nitrous oxide, causing the blood vessels to constrict. What Viagra does is it competitively inhibits the enzyme which prevents the nitrous oxide from getting in. So if we can imagine that this is our enzyme that breaks down the nitrous oxide, our substrate will be the nitrous oxide. Normally we'll go into the enzyme breaks it down, and then the smooth muscle tissue will constrict, getting smaller or narrower. Well, Viagra will block the enzyme by competitively inhibiting it, so the substrate can no longer get in. That means that there's more nitrous oxide left around, enabling the smooth muscle to dilate again or to basically widen. Now, this drug was developed in the hope of dilating blood vessels around the heart to keep blood vessels dilated, keep blood flowing around the heart nice and healthily. But when it went out for human trials, people were coming back saying, sort of, my heart's not any better, but other really interesting things are happening. Because what was actually discovered was that it will also dilate blood vessels in the penis, and particularly the erectile tissue of the penis. So it was enabling men to maintain erections for longer. You know, it's easy for us to snigger about this one, but it's had a really big impact on mental health and the sort of relationships and things like that. So it's been a really important drug, and it just works as a competitive enzyme inhibitor. On a slightly darker note, we have this one, sarin, sometimes referred to as sarin nerve gas or sarin nerve agent. Now, this doesn't occur naturally. This is a complete human-made toxin. It's a nerve agent that's developed, I believe it was developed initially in Germany as a pesticide, but then it was developed further to become a chemical warfare agent. And for those, I don't know if any of you are old enough to remember, but this was actually released by a terrorist organization in Tokyo in the 1990s. It was released in the Tokyo Underground. And it did kill some people and injured quite a few more. And what sarin does is it competitively inhibits enzymes within the nervous system, and basically it will prevent certain nerves from shutting down, so you get overreaction, and eventually it can lead to convulsions and paralysis and death. So it's a really, really nasty nerve agent, but it works again as a competitive inhibitor. So we've got three examples that do very, very different things. Now, one way that you can actually overcome a lot of competitive inhibition is by increasing the amount of natural substrate that we have. So what we are doing with competitive inhibition is we are adding a competitor for access to the active site. So if you increase the substrate concentration, well, now you have more chances of the substrate actually making it into the active site rather than the competitive inhibitor making it in. So by adding substrate, you can actually increase the chances of the normal enzymatic reaction occurring. The other type of inhibition that we've got then is non-competitive inhibition. The reason that this is called non-competitive is because now we have an inhibitor that is not competing for access to the active site like we saw in competitive inhibition, instead, we have a different part or a different site on the enzyme and our inhibitor. So we can see over here, the yellow part here is the inhibitor. The inhibitor will bind to the enzyme and it causes a change in shape or conformity of the active site, and it actually prevents the enzyme from fitting into the active site. So we're not competing for access to the active site anymore. We're actually changing the shape of the active site, preventing the substrate from entering. So you can see that written here. The inhibitor binds at a different site to the substrate, rendering the enzyme substrate complex inactive, or by completely distorting the enzyme so that the substrate cannot bind at all. So because this actually changes the shape of the active site, increasing the substrate concentration won't make any difference because the enzymes are completely distorted. So the active site is rendered inoperative or useless. So you can add as much substrate as you like, and it's not going to be able to fit into the active site. And a couple of examples of drugs or toxins in this case that will impact on will work non-competitively are mercury and cyanide. And these are really, really nasty toxins that affect all manner of different processes within the body. And they work by non-competitively inhibiting enzymes. So let's watch this animation then, which covers competitive and non-competitive inhibition.
During the normal enzyme catalytic cycle, the substrate encounters an enzyme with a specific active site to which it binds, forming an enzyme-substrate complex. The enzyme then facilitates the breakdown of the substrate to its products, which part from the enzyme, leaving the active site free to catalyze another substrate as the cycle begins again. Competitive inhibition occurs when an enzyme encounters a blocker, which mimics the properties of the substrate and binds to the enzyme's active site. Thus, when the substrate is encountered, the active site is not available for attachment and no reaction will occur. Non-competitive enzyme inhibition involves the binding of a blocker to the enzyme away from the active site. This binding causes a conformational change in the enzyme, altering the shape of the active site, which prevents the substrate from binding. No reaction will occur as long as the non-competitive blocker is bound to the enzyme.
Hopefully that helped to understand inhibition then. So let's just do a quick recap of what we've talked about so far. Enzymes can be denatured by heat. If the temperature rises above the optimal temperature, then the enzymes will denature. And they can also be denatured by the wrong pH. And the example that I've given you as we've gone through is coral bleaching. So here we can see healthy coral bleaching healthy coral on the left, and on the right we can see examples of coral bleaching. And we can talk about the coral in a bit more detail in a later lesson. But coral has a pretty complex life, and it doesn't take a lot to upset it. So raising the temperature or changing the pH will certainly do that. One thing that we haven't really talked about yet, but if there is too much substrate and not enough enzyme, the active sites can become saturated. And it kind of makes sense, doesn't it? If you've got 10 enzymes and you've got 10 substrate molecules, well, then the enzymes can operate on one substrate each, and it happens very, very quickly. But if you've got 10 enzymes and you've got 1 million substrate molecules, well, then you end up with this backlog of substrate waiting to get processed. And if that happens, that's what we would say, or when we would say, the enzyme has become, or the active sites have become saturated. And the only way you can overcome that is by producing more enzymes. And this can happen in different stages of life because of different reasons. Whether it's through ingestion of food or the production of waste products, things like that. And we've also talked about the fact that enzymes can be inhibited by drugs and toxins. Some are good, so things like we use them to our advantage, so antibiotics, but also some can be bad. So we talked about sarin nerve gas and gave examples like mercury and cyanide. So let's just quickly recap the take-home messages then that I'd like you to remember about enzymes. So enzymes are catalysts. Enzymes are proteins. Enzymes work by using induced fit to hold the substrate. Enzymes are affected by extremes of heat and pH. Enzymes are big, yet they have small active sites, and the active sites are where the reactions take place. Enzymes can be switched on and off through allosteric control. But enzymes can also be inhibited, so switched off due to drugs and toxins. Enzymes can and do determine species geographical ranges. This is obviously a big issue now with climate change. So here we've got some recap questions then, just to see what you remember. So as per usual, I'll read through the questions, then we can pause the video, and then we'll go through the answers. So number 1, how do extremes of pH affect enzyme structure? Question 2, how does competitive inhibition work? Number 3, how can enzymes affect the geographic range of species? And number 4, give an example of a drug or toxin that works by inhibiting enzymatic reactions. So have a go answering those, and then we'll go through the answers together. Okay, welcome back. Hopefully you've had a chance to answer those questions. So let's go through the answers. So number 1, how do extremes of pH affect enzyme structure? Well, they'll be denatured, they will unravel, and they'll be no longer able to work. Number 2, how does competitive inhibition work? Well, it's when a molecule binds to the active site and prevents the substrate from entering it. So there's lots of different ways that you could have written that, but the key thing here that I wanted you to get to was that competitive inhibition is when a similarly shaped molecule gets into the active site and prevents the proper substrate from getting in there. So number 3, how can enzymes affect the geographical range of species? Well, organisms will migrate to new environments that have the right temperature and pH, or they have to adapt, or they will die. So hopefully you've mentioned something around that, or you might have looked at it from a different range where the temperature and the pH has to be of the right value for the organism to survive by maintaining optimal enzymatic reaction rates. Okay, so number 4, give an example of a drug or toxin that works by inhibiting enzymatic reactions. Well, we looked at some examples, so penicillin, Viagra, sarin, mercury, and also cyanide. But one that I've put there that I haven't mentioned previously. So, but if you know about proteins and enzymes, you might have put there. So it's a little bonus point if you got this. Is that some snake venoms appear to work through inhibiting enzymes as well. So there we've got 4 questions. Hopefully you did okay with those. So I'm just going to show you another quick video. It got ahead of itself there. So I'm just going to show you another quick video which runs through a quick recap of what we've discussed so far.
Enzymes are proteins which catalyse metabolic reactions involving the conversion of a substrate into a product. Enzymes bind with substrates due to their complementary shape. This is called the lock and key model. Rather like a spaceship docking into a space station. In the induced fit model, the enzyme changes shape to accommodate the substrate. Enzymes enable metabolic processes to proceed rapidly at room temperature by providing an alternative reaction pathway of lower activation energy. Without enzymes, these reactions would happen too slowly to sustain life, or would require extreme conditions of pH or temperature. Carbonic anhydrase catalyzes the following reaction between carbon dioxide and water. This reaction proceeds 10 million times faster with the enzyme catalyst than without. A water molecule is coordinated to a zinc atom in the protein. A proton is removed, leaving a hydroxide ion. Carbon dioxide binds to the hydroxide ion on the zinc, and the product is released. When a similar shaped molecule competes for the active site, it reduces substrate activity. This is called competitive inhibition. Adding more substrate increases activity. In non-competitive, allosteric inhibition, a molecule binds to a remote part of the protein, preventing substrate binding by altering the shape of the protein.
Hopefully that made sense. If you need to go back over everything, then by all means do. It can certainly help to go over these complex topics a few times. But what we've covered in this session then, so we talked a little bit about what catalysts are, we talked a little bit about enzymes and what they are, and then we've looked at enzyme structure, we've talked about lock and key hypothesis and the induced fit model, and we've also talked about enzyme inhibition, and I've given you some examples as we've gone through about how enzymes can dictate the geographical range of species. So hopefully that was interesting and helpful, and I will see you in the next session. Thanks very much.